All right, hello, and welcome to lecture number 2 about anatomy and physiology. Today we're going to cover atoms and molecules, and that is chemical basis of anatomy and physiology from a biological perspective. So as far as this is concerned, when we talk about the chemistry of anatomy and physiology, we have biomolecules that we have to discover and see their different aspects as far as how they impact our lives. So in our introductory lecture, we did make mention of a couple of these concepts, atoms and molecules. Here we're going to dive deeper into them for lecture number 2. So we mentioned the subatomic particles previously. We had the electron, the proton, and the neutron. As we can recall, the electron has a negative charge, the proton has a positive charge, and the neutron has a neutral charge. The electron's charge is very small. The electron's mass is very small compared to the proton and the neutron. This means that it's negligible as far as the mass is concerned. So as we can tell here, the relative mass is 1,836th of a proton or a neutron. When we see an atom on the periodic table, we see the element, that is the symbol. We see the atomic number, that's the amount of protons and neutrons that exist in the nucleus. And then we have the atomic mass, which is the total mass put together of all of the particles. So we see the atomic number of 1 here. For hydrogen, that means there is 1 proton and 1 neutron in the nucleus. There's also 1 electron. The electrons are encompassed in an orbital shell around the nucleus. We can see the nucleus containing the protons and neutrons. Around that nucleus, we can form 3 orbital shells. The first one is going to have 2 electrons, the second one can have up to 8, and the third one can have up to 8 as well. We see the nice animation there of our electrons circling around the nucleus. So, when we change the chemical formula of an atom, we form a couple of different things. In order to form an isotope, we have to change the amount of neutrons that are present. So usually the amount of neutrons is going to be equal to that of the amount of protons and electrons. Here in the case of carbon, we have 6 neutrons, we have 6 protons, and we have 6 electrons. If we were to change the number of neutrons to 7, now we have 7 neutrons. That is not going to be equal to 6 protons and 6 electrons anymore. So the atomic number is now going to be 13 instead of 12. Carbon 12 has 6 protons and 6 neutrons, equaling 12. Carbon 13 now has 7 neutrons. If we want to take that even further, we can go to carbon 14, which now has 8 neutrons. In order to get that back down to carbon 12, radioactive decay has to take place. So the neutrons have to slowly be taken off over time, and that is going to be a half-life. So isotopes, the similarities, going to be protons and electrons. Differences is going to be the amount of neutrons that are there. And since the amount of neutrons changes, that means that the atomic mass also changes, because we have added a molecule that is equal in weight to a proton, and we have changed the atomic mass. As far as isotopes are concerned, we use isotopes in things like medical imagery, diagnostics, system-wide functionality, visualization. If we have too many neutrons, which we have in the case of carbon 13 and carbon 14, those neutrons are going to be released. And as those neutrons decay and leave, they produce energy. Energy can come in very different forms. We can have things like X-rays, gamma rays, beta particles, alpha particles. All right, a lot of different radiation can come. That radiation is going to be based off of a time constraint, and that time constraint is called a half-life. A physical half-life of an isotope is going to be the amount of time it takes to degrade 50% of that atom to a stable neutron number. So let's say we have 10 carbon 13 atoms. The amount of time it takes for 5 of those to now become carbon-12 again is going to be the physical half-life. Biological half-life is going to be how long it takes for your body to clear it. So how long it takes the body to clear 50% of that particle weight or particle mass from the body. Radioisotopes in medicine. Here we have a tumor. This is a large beta cell tumor. Here we have used a radioisotope to look at the metabolic rate of a specific area. And the metabolic rate of this area here is much higher than the tissue around it, indicating that we have an abnormally fast-growing tissue area. The radioisotopes inside of there are being degraded at a much faster rate, and that is why it is giving off fluorescence or giving off energy at a much higher frequency, and that causes it to light up and show us that. So for reference, this is going to be a view of the chest cavity. So we have the thoracic cavity here. You have the anterior portion, the posterior portion. So this is going to be a large beta cell lymphoma on the thymus, which is going to be in the mediastinum. All right, so our electrons, the electrons are going to be going around in shells. The amount of electrons that we have is going to be equal to the amount of protons and neutrons if we have a stable atom. The outermost shell contains what we call valence electrons. And the valence electrons are the ones that actually do something. They will either form bonds with other molecules or they will give off energy as they are released. Our first electron orbital, the first shell, can contain 2 electrons. The second one can contain up to 8. And our third shell can contain 8 and even more if we alter it in a chemical lab or force those electrons to stay in the orbital. Different atoms like to have what we term octets or full shells. So 8 electrons in a shell is what we are going to look for as far as the valence shell is concerned. And those electrons are going to do things like bind other molecules and release their electrons to other substances to try and get to that 8 number. So we have 2 in the innermost shell, and we have 8 in the second shell. Common elements that we look at as far as atoms are concerned in biology are going to be that which we find most in the atmosphere and the most in our bodies. We have things such as hydrogen, carbon, nitrogen, oxygen, sodium, magnesium, and chlorine. Now notice how a couple of these have odd numbers. So they have an odd number of protons. That means that things are going to have to go and bind to other substances in order to create an octet or a stable compound. So usually in the atmosphere, atmospheric hydrogen, atmospheric oxygen, atmospheric nitrogen are going to be circulating around as 2 atoms bound together. Hydrogen has 1 proton. That means it has 1 electron. If we are to bind a hydrogen to another hydrogen, it now has 2 electrons, which now fill the first orbital. Now it is stable. If it was not stable, it would be highly reactive and would react and cause explosions all over the place in the atmosphere. Atoms are expressed by formulas. The molecular formula is going to be that which shows simply the types of atoms that are there. So here we see that we have a carbon, an oxygen, and a hydrogen. We have 3 carbons, 3 oxygens, and 6 hydrogens here. This is the molecular formula of glucose. Now, the structure formula shows you how it looks, right? How it looks in 3-dimensional space. So we still have the same amount of atoms. We just have how they are arranged in their specific 2-dimensional structure outline. So we have a 3-carbon chain in the center. We have hydrogens bound to those carbons. And then we have the hydroxyl group bound to these 2 carbons. These 2 structures right here are termed isomers. They have the same molecular formula, but they are different in their structural formula. Next, we have an ionic bond. Ionic bonds are bonds that take place between charged or polar molecules. If something is positively charged, it will try and find something that is negatively charged to bind to. We can think of the magnet example. This allows for those two to bind together and form a neutral compound, which is now stable. So a normal atom is going to have no net charge. It is going to have the equal number of protons and electrons. A cation is that which has a net positive charge. So here we have one more proton than we have electrons. An anion is one that has a net negative charge. In this case, we have one more electron than we have protons. If we put a cation and an anion together, they will bind. And when they bind, they now have an equal number of protons and an equal number of electrons. And that will allow it to be a net zero charge. So we see the positive and the negative here. They will attract one another, bind, and form a stable compound. And this is an ionic bond between a cation and an anion. Normal ionic bonds that we find, one example is going to be salt. We have sodium, which has a net positive charge. We have chloride, which has a net negative charge. Sodium chloride is going to bind together. And once that binding happens, we are going to have a compound that has formed an ionic bond. It is now going to be a solid and is going to form a lattice structure. So we see a salt here that has formed a lattice work and is put together, right? Next kind of bond is a covalent bond. Covalent bonds are going to be between like molecules. So they share electrons, right? Two or more types of atoms that are bonded are going to be covalently bonded. In this case, we have like atoms that are bonded here. In a single covalent bond, we have two hydrogens that are sharing their electrons. In my example earlier, I stated that hydrogen in the atmosphere, if it was left alone, would be reactive because it has a negative charge. However, since we have both electrons here, the inner orbital shell is now full, so it is now stable. And that only requires sharing of one. So now we have a single covalent bond. In a double covalent bond, in the case of oxygen, we are now going to share two electrons. That gives us a stable 8 electrons in the outer valence shell. This forms a double bond, where we are sharing two pairs of electrons between the orbital shells of the two molecules. For a triple covalent bond, we are going to share between two nitrogens. And those nitrogens are going to form a triple bond, which we are sharing 3 electron pairs between. This is to give a stable conformation. Each nitrogen has 7 electrons circulating in its outermost shell. So that means we have to share 3 pairs in order to get to a stable configuration. Now, covalent bonds can either be polar or nonpolar. In a nonpolar substance, it is 2 non-charged particles that are going to share evenly. Each one of their charges does not overtake the other, and therefore there's equal distribution of force between the two. If it is polar, it is going to be an uneven sharing of distribution. One of the molecules is going to be far more attractive than the other and is going to have a stronger pulling force. In order to determine this, we have a method known as electronegativity. Electronegativity is how much pull a certain atom has towards itself when trying to bind to others. So the electronegativity is determined partly by the amount of electrons that are in the valence shell, how close it is to filling that octet rule, and also what other molecules it's binding to. So here, electronegativity of hydrogen. Electronegativity of hydrogen is very low, partly because it only has 1 electron, and partly because it is very small. Oxygen is going to be the largest electronegativity of this group. between carbon and nitrogen, hydrogen and oxygen. Electronegativity is how we get the bond angle of water molecules. So oxygen, having a high electronegativity, is going to pull and tightly bind to two hydrogens, and it's going to form this bent angle. We are sharing an electron between hydrogen and oxygen for both of those molecules. This is a polar covalent bond. So this polar covalent bond is made because the attraction of oxygen to the electrons is very high, and therefore it pulls and tightly binds these two hydrogens. Here's a chart of electronegativity. So electronegativity by definition, the tendency of an atom to attract a bonding pair of electrons. At the top of the electronegativity scale is fluorine. It has the highest electronegativity because it is the smallest and it has the closest setup to the octet rule. This last group here are the group of noble gases. And those noble gases are going to have a full octet. Having a full octet is desirable. So fluorine wants to gain one more electron so it can become helium, and it can now be a noble gas. So the relative electronegativity of an atom is very important to determine how strongly or how weakly it's going to be able to bind to a certain element. See that a hydrogen, even though it's very small, still has an okay electronegativity. 2.1 is the number. So it's still somewhat electronegative. That is because it only has one electron in its entire structure, which means that it needs a second one to become stable. So even though it is very small and it is not terribly polar, it is strong for its size. So its attraction to other molecules, especially those that are not gaseous, is going to be large. All right, so we've discussed our polar covalent bonds. We have unequal sharing there. Our next type of bond is known as a hydrogen bond. Hydrogen bonds are transient. They are somewhat weak, and they happen between various molecules because of hydrogen's high binding affinity. Hydrogen is a very promiscuous molecule. It tries to find other electrons to bind to all the time because it is constantly looking for that second electron to fill its valence shell. So for instance, here if we have a bunch of water molecules together, water molecules are able to bind and dissociate and bind again so quickly because they are composed of all of these hydrogens, and all of the hydrogens on these water molecules continuously bind and dissociate and bind and dissociate again over and over. And that allows for water to maintain a liquid structure, but also not to become gaseous because the attraction is still enough to where the liquid is maintained. Another attraction is glucose. We've heard probably before that water follows glucose, or sugars are attracted to water. And that is because the hydrogens on the glucose molecule will very readily bind to water molecules around it. They will very easily find those hydrogens, and we will have hydrogen bonds transiently form between water molecules and glucose molecules. So in order to see this different functionality, for our salt structures, salts are going to form between a positive and negative cation and anion. For example, here we have sodium chloride, standard table salt. If we are going to put this into water, if we have water, which is a universal solvent, and we have something like salt being the solute, to form a solution here, it's going to be pretty easy because salt will dissociate rather quickly in water. The hydrogen bonds will quickly dissociate the two, sodium and chloride, and they will encompass those atoms. Salts are usually hydrophilic substances, meaning that they like water or they attach to water easily. They're going to be electrolytes, which means that they are able to conduct electricity, and they're going to form hydrogen bonds between the water molecules to dissociate pretty evenly throughout the solution. Now, a polar substance, such as sugar, is also going to be able to dissolve, but it's going to have a lot more water associated with one molecule. So that means that a molecule of glucose is not going to dissolve completely. For salt, we had sodium and chloride dissociate. They become individual atoms of sodium and chloride. Here for glucose, it dissociates into individual glucose molecules, but it does not get any smaller than that. It goes down to an individual glucose, and the water will now hydrogen bond to the hydrogens located in the structure of that glucose, but it does not break. So it's hydrophilic, it likes water, but the smallest unit of glucose does not dissociate and become individual atoms. If it is nonpolar substances like fats are usually not going to like dissolving very easily. So for this instance, we have a hydrophobic substance, mainly a lipid. A fat or a lipid is going to be a nonpolar substance, and it is going to maintain what is known as hydrophobic exclusion. It repels water, it does not like to dissolve in it, and the binding between the nonpolar molecules maintains itself very strongly. So the water molecules are not able to wedge in and dissociate them. An amphipathic molecule is one that has both hydrophilic and hydrophobic properties. Something like a phospholipid, what we find in cell membranes. Cell membranes are composed of phospholipids. We have a phosphate head group, which is going to be composed of a phosphate and glycerol. And we have two fatty acid tails composed of fatty acids, which are lipids. Now, as we remember, lipids are going to be nonpolar substances that are hydrophobic. So they do not like water. So they're facing inward away from that water. The hydrophilic head group of the phosphate head is going to be liking water. So it's going to face either side. And this is how we have the membrane bilayer structure that forms between the two layers. Now, if we look at a solution, for example, here we have the solution of blood. Blood as a solution is going to be carrying around things that are completely dissolved, partially dissolved, or not dissolved at all. So blood is containing our universal solvent, which is water. Molecules that are dissolved to a great degree or pretty much entirely dissolved in solution are going to be things like salts, sugars, and very small nutrients. One that's going to form a colloid, which is going to be small particles that are still present and are able to scatter light, are going to be medium-sized molecules like proteins. And molecules that are going to form a suspension are going to be the formed elements of the blood, the red blood cells, white blood cells, and platelets. These are large and clearly visible in the blood, and they will settle towards the bottom of a solution as they precipitate out. So in a solution, we have pure dissolving, no settling out, and there is no scattering of light. In a colloid, we have scattering of light, but it does not settle out. So it's very fine, misty particles that still float around. And then for a suspension, we have large particles that do not dissolve. And if they are centrifuged down, they will settle out as a precipitate. So what chemical property connects most substances in the blood? And that chemical property is that most substances are going to be ionic in nature. So blood and pH. pH is the measurement of the acidity or alkalinity of a substance. If something is acidic, it has a low pH. If something is alkaline, it has a high pH. The pH scale runs from 0 to 14. Something that is 0 is going to be very acidic. Something that is 14 is going to be very basic. Human blood is normally around 7.4, which is close to neutral. Neutral is 7, and 7 is based off of the amount of hydrogen ions that is located in pure distilled water. So everything is around that 7 number. When we find other things that we take in on a normal basis, such as juices, um, urine, we have excretion of milk, saliva, those are going to be acidic compounds. On the basic side, things that we use on a regular basis, something like bleach or something like ammonia or dish cleaner, those are going to be basic substances, mainly going to be used as household items. If we undergo a period of acidosis, this is going to be where our bloodstream has a pH that is lower than 7.35 for a prolonged period of time. That is going to be an acidotic state. An acidotic state is going to produce death usually if the pH goes below about 6.8. If we are alkalotic or alkaline blood is going to be when the pH goes above 7.5 for a prolonged period of time. An alkalosis that is above 7.8 or so for any period of time will usually result in death as well. So what are our organic molecules? Well, the organic molecules that we utilize are going to be lipids, which are fats, nucleic acids, which are used in DNA and RNA, protein, which we use as a building block for all tissues, and carbohydrates, namely glucose, that we use for cellular energy. Some examples of that going to be a bagel for carbohydrates, going to be animal fat for lipids, going to be nucleic acids such as adenine for DNA, and protein, we have a steak here. So organic molecules usually have these substances, all right? Organic molecules will always have atoms of carbon and hydrogen associated with them. Usually we can tack on oxygen as well. Now they may also have nitrogen, phosphorus, and sulfur, but they will always have carbon and hydrogen. Remember, we are carbon-based life forms, so we always have to have carbon. Biological, organic molecules, beings, things of that nature are always going to have carbon. So here's the functional groups that we normally find. Functional groups in a carbohydrate, it's going to be the hydroxyl group. This is a chemical formula of an oxygen and a hydrogen together, or OH. And a carboxyl group, normally found in proteins. This is going to be a carbon double bonded to an oxygen and single bonded to a hydroxyl. For an amino group, which we usually find in amino acids and proteins, we have a nitrogen that is going to be single bonded to a hydrogen on either side. And for a phosphate group that we normally find in DNA and also in ATP, which is adenosine triphosphate, the main cellular energy source that we get, this is going to be a phosphate that has 4 bonds to oxygen around it and has a net negative charge. In terms of amino acids, an amino acid is going to have a carboxyl group on one end. It's going to have an amino group on the other. Carboxyl group is going to act as a base for that substance. The amino group is going to act as, the carboxyl group is going to act as an acid for that substance. The amino group is going to act as a base. That is why amino acids, for the most part, are going to be neutral, non-polar compounds. Lipids. Lipids are made up of long carbon chains that are attached to hydrogen molecules. If it is saturated, they are all single bonds. If it is unsaturated, there are double bonds present. Lipids are nonpolar substances, so therefore they are hydrophobic. They do not dissolve and they do not form solution with water. The functionality is to store energy. They are also present in hormones like steroids, cholesterol, and they form the cell membrane through the phospholipid bilayer. There are 4 types of lipids that we find in the human body. Triglycerides, which form the storage form of fat, which is adipose tissue. Phospholipids, which form the cell membrane. Steroids, which form the steroid hormone groups. And eicosanoids, which are immune cells and work in inflammation. So triglycerides is our storage form. We have a glycerol head group and 3 fatty acid tails. When we create fatty acid storage, we form a triglyceride by binding the 3 fatty acid tails to the glycerol head group. Triglycerides are the main component of adipose tissue that we find in the body. It's our main storage form of fat. It provides insulation for the abdomen and provides structural support for the internal organs. Top right here, we can see fat cells. Mostly fat cells are going to be open and centralized. All of this white stuff is fatty acid droplets, and the nucleus of the cell is pushed out to the periphery. And we can see that that fatty acid droplet is going to be pushing the nucleus to the edge of the cell, and it's going to grow larger and larger as more fat is stored. Now, triglycerides are going to be saturated or unsaturated. Saturated fat is going to be one that has all single bonds. So we have a carbon chain here with all single bonds. If we have an unsaturated fat, it's going to have a double bonded structure. Double bonded structure is going to have two carbons that are linked by a double bond in the chain. Unsaturated fats are going to be named by how many double bonds they have. A monounsaturated fat has one double bond. A polyunsaturated fat has multiple. And the name of those fats is going to be where that double bond exists in the chain. So for example, we have oleic acid here as the monounsaturated example. That is an omega-9 fatty acid. Omega-9 is going to be right here at the 9th carbon. So we have the omega carbon, the last carbon, subtracted 9. So omega minus 9. So if we take the omega carbon and we subtract 9 carbons to the center, here we have a double bond in the middle. Same thing for omega-6. Here, we take the last omega carbon and we subtract 6, and we find the first double bond. For linoleic acid, we have omega-3. Take the last carbon, we subtract 3. Now we have 3 areas of double bonds between the carbons. Phospholipids are going to be making up the cell membrane. Cell membrane is going to be comprising a phospholipid with a phosphate head group and fatty acid tails. This is an amphipathic molecule, if we remember. So the head group is going to be hydrophilic, likes water. The tails are going to be hydrophobic, so they will be facing inward towards the towards the inner portion that is dry. For our structure, we have a phosphate linked to a glycerol. And instead of a 3 fatty acid tail like we have for triglyceride, we will have 2 fatty acids in the tail group. So there's an organic group formed by a choline, an ethanolamine, and a serine. A phosphate linked to a glycerol and 2 fatty acid tails. This forms the lipid bilayer of cell membranes. It is selectively permeable. It will allow fat-soluble substances through, and it will reject ionic substances and water-soluble substances. Next categorization is steroids. Steroids are all based on a cholesterol structure. So they all have this 4-ring structure base. This is our standard cholesterol molecule. From that cholesterol, we can derive many different hormones. We can derive testosterone, we can derive estrogen, we can derive cortisol. Steroids are going to be in the form of hormones, in the form of cholesterol, or in the form of bile salts, absorbing fat through the intestine. Cholesterol is also a vital component of cell membranes. They are lodged within the cell membrane phospholipid bilayer, and they will increase the fluidity of that cell membrane, so it allows it to move around more freely. Finally, eicosanoids. These are going to be 20-carbon fatty acids, and these are immune functionality. Eicosanoids are there for inflammatory responses. They're mainly synthesized from arachidonic acid and will create compounds known as prostaglandins, thromboxanes, and leukotrienes. Some other lipids in nature. We have several vitamins which are lipid-based. Vitamin A, vitamin E, vitamin D, and vitamin K. These are fat-soluble vitamins, so therefore we can store them within fat in the body. Here we have a glycolipid. A glycolipid is one that is attached to a lipid residue. We find these a lot on cell membranes, as we have sugar and glycogen molecules attached to the surface of a cell membrane, and that is a glycolipid structure. Carbohydrates are named because they are hydrated carbons, meaning that we have water associated with a carbon. That makes sense because we have the affinity for water so tightly built in with carbohydrates. So we have a standard 5 or 6 carbon ring. We have a hexose sugar in glucose, hexose meaning 6, so we have a 6 carbon ring there. A monomer is a single sugar, which is the basic building block of a sugar carbohydrate compound. And we have polymers, which are many monomers linked together. A disaccharide is going to be 2 sugars, and a polysaccharide is 3 or more. A monosaccharide is going to be a single sugar. So, some sugars that we find in the body. At our base structure, single ones we find are glucose. Glucose is our base structure of sugar and is going to be utilized by cells to the highest degree. Also, we find things such as galactose. We find fructose. These are nutrient-based sugars. These are hexose sugars. For pentose sugars, pentagram meaning 5, these are found in DNA and RNA. We have ribose for RNA, ribose sugar, and we have deoxyribose found in DNA for deoxyribose sugar. Disaccharides that we find are going to be the ones such as sucrose, lactose, which we find in milk sugar, and maltose. Now, all these are going to be broken down eventually into glucose if they are utilized as cellular energy. If we are going to store that energy, we will form glycogen, which is the process of glycogenesis. If we want to use energy, we will use it from that glycogen. We will break those bonds and get a glucose out of it. That happens in the liver and in skeletal muscle. So we eat sugars, we eat carbohydrates in the form of starch, cellulose, whatever else. Hydrolysis is going to yield glucose once we go out of the digestive tract. Then in the liver, we will decide what we're going to do with it. We're either going to store it by making more glycogen, or we are going to use it when we need energy for our cells to create metabolism. So to make a polymer, the standard is the process of dehydration synthesis. This is a condensation reaction, meaning that we're going to lose a water molecule and form a dimer between 2 monomer substances. So we're going to lose 2 hydrogens and an oxygen, which are going to form water as a byproduct. And we will form a bond around a second oxygen, which is left over, to form a dimer. Hydrolysis is using water to break that bond. So it's the breakdown of biomolecules such as glucose. This is where we have water come in, and it's going to break apart that bond of the dimer. And it's going to contribute an oxygen and 2 hydrogens back onto that carbon structure. Nucleic acids we find mainly in genetic material. So here we have it in DNA and RNA. The monomers are known as nucleotides. We have 2 groups, pyrimidines and purines. The pyrimidines are going to be cytosine, uracil, and thymine. Thymine is present in DNA. Uracil is present in RNA only. The purines are a double ring structure, and those are adenine and guanine. Now, in terms of binding, cytosine will always bind with guanine. Uracil will bind with adenine, and thymine will also bind with adenine. In DNA, we will have thymine and adenine binding together. In RNA, we will have uracil and adenine binding together. The basic structure of the nucleotide is going to have a sugar, whether that be ribose or deoxyribose. a phosphate group, and a nitrogenous base. So the polymer of the nucleotides is either going to be DNA or RNA. And these linkages are made by phosphodiester bonds. Phosphodiester bonds join the nucleotides together. For DNA, we have a double-stranded structure. Again, thymine is going to bind with adenine. Guanine is going to pair with cytosine. In the cell, we find DNA in the nucleus. We also find it in the mitochondria. As far as the sugar is concerned, here we have deoxyribose. It is a deoxygenated ribose sugar. Here, if we notice, at the 2' carbon, we have no oxygen molecule, so we have no hydroxyl group here. If we get to ribose and form RNA, we would find that we do have an oxygen here. So that is ribonucleic acid. So DNA is deoxyribonucleic acid. And as mentioned here, we have that structure. So the ribose sugar has now become oxygenated again. So we have ribonucleic acid. Ribonucleic acid is usually going to be single-stranded, whereas in DNA we had a double-stranded structure. RNA's cellular location is going to be in the nucleus and also in the cytoplasm. It communicates with ribosomes in the cytoplasm of the cell, so it will be consistently migrating out of the nucleus into the cytoplasm to work with the cellular machinery. There are 3 main types of RNA. Messenger RNA, which goes to the ribosome, ribosomal RNA, which maintains the ribosomal structure, and transfer RNA, which moves amino acids to and from the ribosome. And again, as I mentioned here, there is no thymine. Here in RNA, we have uracil. So we have uracil binding to adenine. There is no thymine. Other important nucleic acids, we have ATP. This is adenosine triphosphate. Adenosine triphosphate is the main cellular energy source that we use. It is created by 2 mechanisms, whether that be glycolysis or the oxidative transport chain. We have 2 bonds, all right, 2 phosphate bonds here that can be used as energy. Each time that we cleave off one of those phosphates, we release energy from that bond. And when we release energy from that bond, we can use that for cellular processes. So adenosine triphosphate becomes adenosine diphosphate for 2. And when we do that, we release the energy from that 3rd phosphate. And we can use that for our cells. All right, proteins. For proteins, we're going to have several different functions. We can have regulation, such as in insulin. We have storage, such as ferritin, which is iron. We have enzymes or catalysts, such as DNA polymerase. We have defense in the terms of immunoglobulins. We have transport in terms of hemoglobin, glucose transporters. Structural support, collagen and elastin fibers. And movement, contractile proteins of the skeletal muscle, actin, and myosin. Proteins are made up of monomers, and those monomers are our traditional amino acids. We have an amino group on one side. Again, remember that acts as a base. We have a carboxyl group on the other, acts as an acid. This is going to form a non-polar stable compound. The R group is what changes. The R group is the functional group. To determine the amino acid that we have, we are going to be able to use the R group. A polymer is going to be multiple amino acids put together. An oligopeptide is going to be between 3 and 20 amino acids. Polypeptide is between 21 and 199. And if it's above 200, it is a full-fledged protein. To form polymers, we use dehydration synthesis, just like we used for the carbohydrates. They form peptide bonds between the amino acids. Similarly, to break it down, we use hydrolysis, just like breaking down the carbohydrates. We find glycoproteins on cell membranes. Glycoproteins are proteins that are attached to carbohydrates. So here's an example. Here we have dehydration synthesis. So we have an amino acid, and we are forming a peptide bond between an amino acid. We are releasing a water molecule and then forming a peptide bond to form a monomer and monomer bond. So this is a polymer protein. We still have an amino group and we still have a carboxyl group. So that means that we have a neutral charge. And we have peptide bonds between each individual amino acid. For protein structure, we have 4 levels. We have a primary amino acid sequence, which is just the amino acids put in line. We have a secondary structure, whether those amino acids are going to form an alpha helical structure, such as this here, or a beta sheet, which is this pleated sheet structure here. We have a tertiary structure, which is what the 3-dimensional shape is in space. So that's putting our amino acids together and showing what the protein looks like in a 3-dimensional orientation. Finally, we have the quaternary structure. If we have multiple proteins that we're putting together, the combination of those subunits is going to form a full protein molecule, which is now a quaternary structure, or what it looks like in 3-dimensional space. Some intermolecular interactions that we find. For proteins, we're going to find hydrogen bonds. We're going to find hydrophobic or van der Waals interactions. We're going to find ionic bonds, and we're also going to find disulfide bonds. So we have several different bond types that we can have between amino acids to form a protein. To denature a protein, we have to change its chemical layout. To do this, we have to change the conformation. And we can use several different things to do so. We can vastly increase or decrease the temperature. We can introduce other chemicals. We can change pH or make it really basic or really acidic. And if we are going to interrupt these chemical bonds, we're going to alter the structure of the protein. Usually it is permanent. So usually if we disrupt the activity, it is going to be a permanent change. For example, here we have the egg. If we fry the egg, the liquid white of the protein becomes denatured at a high temperature. It then coagulates, those bonds are broken, and now we have a solid coagulated egg white. So some different protein structures here. All right, we've got silk protein. This is collagen for structural support. We have an immunoglobulin down here in the bottom right. That's immune functionality. We have a retinal binding protein, which we find in the eye. All right, so several different protein structures here. So to recap, the lipid monomer that we have, right? We have lipid monomers that are going to be the individual fatty acids. The polymer is going to be a combination of fatty acids. The structure is going to be either triglyceride, phospholipid, or the diacylglycerol. Special bonds between lipids are going to be phosphodiester bonds. And some examples are the triglycerides. Carbohydrates, our monomer structure is going to be individual glucose. Our polymers, we're going to put several different ones together, such as glycogen. Structure is going to be either a pentose or hexose ring. Special bonds between this is going to be glycolytic bonds. And examples are things like sucrose and fructose. For nucleic acids, the monomer is a nucleotide. Polymer is going to be things like DNA and RNA. Structure is either going to be an alpha or beta helix or a single strand. Bonds between these are known as nucleic bonds or phosphodiester bonds as well. Examples, adenine, guanine, cytosine, thymine, and uracil. And for proteins, our monomers are amino acids. Polymers are full proteins, oligopeptides, polypeptides are proteins. Structure is going to be between the proteins. Special bond is a peptide bond. And examples are going to be things like we saw with retinal binding protein or collagen. So you can use this chart to fill out on your own, get some different examples going on there. And that is the end of lecture number 2. Hope you all enjoyed it. Thank you so much for watching, and I'll see you again for lecture number 3.